Hello learners, in our earlier unit we have learned about the tannins. Today we will be learning about the most important bioactive component of tea, that is catechins. In this module we will be learning about chemistry, metabolism, bioavailability and most important its health benefit. Catechins are the most active constituent polyphenol in the green tea. Epigallocatechins, which accounts for almost 50% of total catechin content of green tea extracts, which makes about a 30% of dry weight of green tea leaves. Catechins have got strongest chemopreventive potentials properties along with anti-inflammatory, anti-mutagenic, anti-carcinogenic effects also seen with green tea catechins. Catechins are known as a type of polyphenol naturally occurring in certain foods and medicinal plants such as legumes, stem bark of Fabia spices, tea, that is mostly green tea, buckwheat, grapes, cocoa beans, lychee and apples. Catechins have been regarded as a characteristics compound in green tea for a daily beverage and a crude medicine in Asia, especially in China and Japan for thousands of years. In general, catechins mainly include catechin, epicatechin, epicatechin gallate, epigallocatechin and its stereoisomers gallocatechin, epigallocatechin gallate and its stereoisomer gallocatechin gallate, with their composition being similar to each other. So let us now learn about the chemistry of catechins. In terms of chemical structure, polyphenols are compounds which have different amount of phenolic rings attached by two or more hydroxyl groups, resulting in more than 8,000 dietary polyphenols, which have thousands of chemical structure ranging from simple molecule to highly polymerized compound. Catechin and its stereoisomer in cis or trans configuration with respect to carbon 2 and 3. Epicatechin cis and catechin trans are flavin 3-OL compounds. Through esterification with gallate group, flavonols can form epigallocatechin gallate. The gallic acid esters of epigallocatechin at position 3. Monomeric catechins can further react and synthesize condensed catechins which are produced by random polymerization. Let us now learn the metabolism and bioavailability of catechins. Catechins can be absorbed by gastrointestinal tract as catechins themselves, and their metabolites are mainly formed in the acidic environment of stomach. Enterohepatic recirculation, the mucosa of small intestine and microbial metabolite in the colon. All the catechins may undergo three types of metabolic pathway: methylation, glucuronidation and sulfonation in the liver and intestinal tissues in humans, like epicatechin transferrin into plasma itself or corresponding glucuronide sulfate methylated metabolite produced by uridine 5 diphosphate glucotransferase, sulfur transferase and catechol O methyltransferase enzymes. Human study has revealed that after the absorption in small intestine, the peak plasma concentration of epicatechins and epigallocatechin glucuronide and their conjugated metabolite would be reached, varying from 1.6 to 2.3 hours. Then the plasma concentration rapidly decreases due to short apparent half-life, while few dietary catechins and related phenolic compounds are not changed except epigallocatechins gallate in 3 plasma. When combined with different polyphenol, catechins may show a very different bioaccessibility too. Owing to existence of the material such as sucrose, ascorbic acid, which may maintain the stability and protect from degradation in alkaline condition. The bioaccessibility of catechins has been regulated. This may explain that in the rats, the chocolate including high sucrose levels enhance the plasma concentration of catechin metabolite than milk and dark chocolate. It has been found that epigallocatechins gallate and some metabolites can strongly inhibit methylation. Let us now understand the sources of catechin. High concentration of catechin can be found in red wine, broad beans, black grapes, apricot and strawberry. And of course, epicatechins concentration are also found in apple, blackberry, broad beans, cherries, black grapes, pear, raspberry and chocolate. Finally, epigallocatechin gallate and epigallocatechin gallate are found in high concentration in both black and green tea. Green tea contain high concentration of 4 main type of catechin. The green tea catechin are epigallocatechin gallate, epicatechin gallate and gallocatechin. Some of these catechin including epigallocatechin gallate are found solely in tea. On an average, catechin make up around 25% of green tea leaf composition. Catechin content of different types of tea depends largely on where and how tea is produced. Tea catechins are present not only in leaves but in the herbal varieties as well. These tea catechins are released when tea leaves are infused in hot water. Let us now understand the health benefits of catechin. Catechin is an antioxidative agent. Catechin possesses both direct antioxidant effect and indirect antioxidant effect. The catechin can scavenge free radicals and chelate redox active metals, making them direct antioxidant. At the same time as an indirect antioxidant, catechin regulate protein synthesis activities and signaling strategies such as mediating the property of pro-oxidant enzymes. The results of number of laboratory experiments have demonstrated that the benefits of green tea and catechins propose the mechanism of action. The target of gallocatechins include abnormal accumulation of fibrous protein such as alpha, beta and gamma synuclein inflammation, elevated expression of pro-apoptotic proteins and oxidative stress, which are associated with neural cells dysfunction and death in cerebral cortex. The findings have suggested that the potential use of green tea have shown prevention in treatment of neurodegenerative diseases and should be useful for development of new drugs. There are several other mechanisms by which catechins and other tea components exhibit anti-cancer activity too. This include antioxidative effect, inhibition of cell cyclic progression, inhibition of nuclear factor, activation of mitogen activated protein kinase cascade, and epigenetic modification. Epigallocatechins, gallate, enriched catechins and green tea infusion have been shown to inhibit metastasis, possibly by inhibiting metalloproteinase activity. It was shown that oral administration of green tea catechins resulted in almost complete inhibition of distant site of metastasis of prostate cancer cell. Studies using cultured cells and laboratory animals have demonstrated anti-diabetic activity of green tea and catechins. They exert anti-diabetic activity by inhibiting the alpha amylase and alpha glucosidase activity, inhibiting glucose absorption in small intestine, protecting the pancreatic beta cells, improving insulin sensitivity of peripheral organs, and inhibiting gluconeogenesis from non-carbohydrate source such as amino acids in the liver. Green tea has also shown and catechins have also shown its impact in liver disease. In galactosamine induced hepatitis models, the rats that consumed green tea beverage showed a reduced hepatic gene and blood protein expression of pro-inflammatory cytokines such as tumor necrosis factor and interleukin factor. Catechins such as epigallate catechin gallate are particularly powerful in preventing heart disease. Catechin also prevents inflammation and reduce the platelet clumping, helping to reduce the risk of blood clot and heart attack. Catechins were also found to regulate blood pressure. Let us now conclude, learners. Green tea contains various components with specific health promoting effect, and it is believed to have protective against diseases such as cancer, obesity, diabetes, hepatitis and certain neurodegenerative diseases. Majority of scientific evidence based on cellular and animal experiments as well as number of human epidemiological and interventional studies indicate that green tea and epigallate catechins gallate have beneficial effect against various diseases. However, conflicting results have been reported since the confounding factors could affect the results. The future studies should eliminate such fractions to better understanding the benefits of green tea for human health. Genetic, environmental factors such as race, age, lifestyle, intestinal microbiota may also influence the result observed in human studies. Although care should be taken not to ingest green tea and supplement with catechin contents, recent findings suggest that habitual drinking of tea promote longevity.